If you've ever wondered why you can go without eating for a while without collapsing due to low blood sugar, you can thank glycogen. Glycogen serves as a fuel reserve during the early stages of fasting. In this sketch, we'll visit the beloved Sugar Street at Sketchyland to explain how glycogen is made and degraded. Ah, Sugar Street, where all your sugar fantasies come to life. Come watch the cursed castle from afar as you enjoy the 5 C's: cotton candy, churros, cupcakes. Anywho, glycogen is a large, branch polysaccharide that functions as the main storage form of glucose. And it looks just like cotton candy. Glycogen is made, stored, and broken down in the liver and skeletal muscle. We've redrawn the cotton candy machine to look like a liver. In the liver, glycogen helps maintain normoglycemia during the first 12 to 18 hours after eating. See the ghoul's muscles? Skeletal muscle uses glycogen for energy during exercise. Let this billboard for cytosol remind you that glycogen synthesis and degradation take place in the cytosol. Okay, now that you know some general features, how do we make glycogen? Well, as you probably already know, we'll start with glucose, illustrated by the sugar sprinkles. Depending on where this glycogenesis is going down, either hexokinase or glucokinase uses ATP to trap glucose inside cells by making glucose 6-phosphate, or G6P. This step is irreversible, meaning those enzymes can't turn that G6P back into glucose. The baker with a kind button using a P battery to power up that oven is hexokinase. Note the hexagon spatula for hexokinase. Meanwhile, the cupcake vendor with a kind button also replacing the P battery in the P battery oven is glucokinase. All sales irreversible will help you remember that both of these enzymes catalyze irreversible reactions. The product is glucose 6-phosphate, or G6P, represented by the 6-shaped cupcake. Before we move on to the next step, let's mention some key differences between hexokinase and glucokinase. Hexokinase is present in the brain, skeletal muscle, and all other tissues except the liver and pancreatic beta cells. We've made the hexokinase baker super buff so that you can remember that hexokinase is present in skeletal muscle. Hexokinase has a high affinity for glucose, represented by a low Km, and low capacity, represented by a low Vmax. As a quick refresher, to find Km, identify the concentration of a substrate, which in this case is glucose, where the enzyme is working at 50% capacity, or 1/2 times Vmax. The low Km value of hexokinase allows the brain and other tissues to preferentially utilize glucose when there's limited glucose available. The low Vmax value of hexokinase means that the enzyme is fully saturated, or active, during the fasting state. The hexokinase baker adding a giant sticky sprinkle, but just one sprinkle, will help you remember high affinity, low capacity. In contrast, glucokinase differs from hexokinase in several ways. 1, it's present in the liver and pancreatic beta cells. 2, it serves as a glucose sensor. And 3, it has a low affinity for glucose, a high Km, and a high capacity to sequester glucose inside the liver when glucose concentrations are high, or a high Vmax. We've illustrated a liver plate and beta cell phone to help you remember that glucokinase is found in the liver and pancreatic beta cells. What about the candy thermometer? Well, when blood glucose levels and insulin are high, glucokinase will remove glucose from the blood to the liver so that it can be stored as glycogen. Yeah, so pretty much like a sensor. See the glucokinase vendor adding many non-stick sprinkles to the cupcake? That's because glucokinase has a low affinity for glucose, and it can bind to many glucose molecules before getting saturated. You should know that glucokinase is predominantly active when glucose concentrations are high, i.e. after eating a large meal. Okay, so let's talk about inhibitors. As we discussed in the glycolysis sketch, G6P, the product of step 1, inhibits hexokinase via product inhibition. On the other hand, fructose 6-phosphate, or F6P, inhibits glucokinase. Speaking of things falling on heads, the vendor better watch out for that 6-shaped cherry. Alright, let's move to the next step. In step 2, phosphoglucomutase reversibly transforms G6P to glucose 1-phosphate, or G1P. This alien hat vendor lost in his daydreams instead of selling 1-shaped churros is phosphoglucomutase. That reversibly is important, so don't forget it. We'll use this dreamy young gent again to catalyze the backwards reaction when we break down glycogen to get glucose. You're probably thinking, what the heck is a 1-shaped churro? Yeah, it's pretty much like a regular shaped churro, but with like a bent top. For our purposes here, it's G1P. In the next step, UDP glucose pyrophosphorylase uses UTP to turn G1P into UDP glucose. Who wants some fudge dip churros? They're warmed up by ultraviolet P batteries, so you know they're going to be ultra amazing. Okay, now that we've made UDP glucose, we can finally start making glycogen. But first, we need to make a primer. Jeannie, it's your cue. Yo, Jeannie. Jeannie looks like a third-year med student on the first day of rotations. Talk about Jeannie in headlights. Anyways, this clueless Jeannie is glycogenin, an enzyme that creates a short, non-branch glycogen primer from several UDP glucose molecules. After we make the primer, glycogen synthase takes over by linking glucose monomers via alpha-1,4 glycosidic linkages. In other words, glycogen synthase joins carbon 1 of one glucose monomer with carbon 4 of another glucose monomer. Glycogen synthase is shown by the same buff ghoul fixing a cotton candy machine with the screwdriver. How will you remember that glycogen synthase creates alpha-1,4 bonds? It's all in the sugar bags. Glycogen synthase is the rate-limiting enzyme during glycogen synthesis, and like all rate-limiting enzymes, glycogen synthase is highly regulated. All of this candy talk must be making you hungry. Please don't hesitate to grab some cotton candy because you're welcome inside the candy shack. Remember that inside is our recurring symbol for insulin. Insulin is the main hormone that activates glycogen synthesis, but cortisol and G6P are also activators. What about glucagon and epinephrine? Well, as you might expect, they work to oppose insulin by inhibiting glycogen synthase. We are almost done making glycogen. In the last step, glycogen branching enzyme removes alpha-1,4 linkages from approximately every 7th glucose residue and creates alpha-1,6 branches to condense glycogen. The warlock carrying a branchy broom is glycogen branching enzyme. Note the 6 shaped ties hanging from the sugar bags to help you remember alpha-1,6 branches. Excellent, we've made a bunch of glycogen. Now what? Break it down? But I just spent all that time making it. Ugh. Fine. In the first step of glycogenolysis, glycogen phosphorylase catalyzes the rate-limiting step of removing G1P residues from branch glycogen until 4 glucose residues are left on a branch. The man feeding one-shaped churros to the ducks is glycogen phosphorylase. Why did we include a pair of ducks? They represent pyridoxine, aka vitamin B6. Without it, glycogen phosphorylase would be just like my brain after a shelf exam. Muddy and totally useless. Let's briefly discuss how glycogen phosphorylase is regulated. Glucagon, epinephrine, and AMP all activate glycogen phosphorylase. On the contrary, insulin, glucose-6-phosphate, and ATP inhibit glycogen phosphorylase. Please check out our glycogen regulation sketch for more information. Besides glycogen phosphorylase, we also need 2 debranching enzymes to degrade glycogen. After 4 glucose residues are left on branch glycogen, 4-alpha-D-glucanotransferase transfers 3 glucose units to the linkage site. The gardener cutting between the first and second flower of a 4-flower branch is 4-alpha-D-glucanotransferase. Then alpha-1,6-glucosidase, a second debranching enzyme, removes the remaining glucose residue at the branch point. This enzyme will be represented by another gardener cutting the last flower off right at the branch point. In the next step, phosphoglucomutase turns the remaining G1P to G6P. Finally, glucose-6-phosphatase removes a phosphate group from G6P, producing glucose. Just to remind you, glucose-6-phosphatase is only present in the liver. Therefore, skeletal muscle glycogen cannot be metabolized to glucose and can only make G6P. The person removing a P battery from the cupcake oven is glucose-6-phosphatase. And we've finally made glucose once again. Whew! Okay, I hope that didn't give you a sugar coma. Let's wrap up this sketch. Glycogen is the main storage form of glucose that serves as fuel reserve during the early stages of fasting. Glycogen is made, stored, and degraded inside the cytosol of liver and skeletal muscle. In order to make glycogen, hexokinase and glucokinase first irreversibly transfer a phosphate group from ATP to glucose, making G6P. Next, phosphoglucomutase turns G6P to G1P. Then, UDP glucose pyrophosphorylase uses UTP to make UDP glucose from G1P. Glycogen uses several of those UDP glucose molecules to create a small primer. Then, glycogen synthase takes over, linking UDP glucose monomers by alpha 1,4 bonds. Insulin activates and glucagon inhibits glycogen synthase. Finally, glycogen branching enzyme removes alpha 1,4 linkages from approximately every seventh glucose residue and creates alpha 1,6 branches. In order to degrade glycogen, glycogen phosphorylase first uses inorganic phosphate to release G1P residues from branch glycogen. Insulin inhibits and glucagon activates glycogen phosphorylase. After 4 glucose residues are left on branch glycogen, debranching enzyme transfers 3 glucose units to a linkage site to allow another debranching enzyme to remove the remaining glucose residue at the branch point. Phosphoglucomutase turns G1P back into G6P. And finally, glucose 6-phosphatase removes a phosphate from G6P to make glucose.